I believe it is important to talk about the evolution of the eukaryotic cell because these are very complex, or the most complex forms of cells that we find out there. I actually compared on the first tutorial prokaryotes and eukaryotes, and you could clearly see that eukaryotes were more complex than prokaryotes. So I'm writing it down, more complex. But to understand the evolution of eukaryotic cells, there is this theory that is quite important. You probably heard this in biology, in basic biology, but it is called endosymbiosis or theory, or endosymbiotic theory. And it explains great part of the evolution of the eukaryotic cells. And I will go through more detail throughout this tutorial. But the first thing to understand is that to end up with a complex form of life, a complex cell, you have to start with a simpler one. So I'm writing it down. The earliest form of life that you see out there was a prokaryote. Prokaryotes are simple. Bacteria are simple. You see it, that the structure itself is simpler than a eukaryote. Why is that? Well, I can just mention one thing right now. For example, organelles. Organelles are present in eukaryotic cells. For example, membrane-bound organelles. And we are going to talk about the evolution of eukaryotic cells, and we have to mention how these organelles show up. So how peroxisomes, how mitochondria end up in eukaryotic cells. And also, if you want to be a well-rounded student, I think it is important, even though you probably talk more about human cells, we need to talk about chloroplasts too. Just to understand, have an idea of how chloroplasts, these organelles found in plant cells, actually become part of the cell. So I'm going to start with the very first step of the evolution of the eukaryotic cell. And as you know, we started with a prokaryotic cell, earliest form of life. So this is the first step. And I have my prokaryotic cell, a bacterium here, very poorly drawn, but it illustrates the basic structures that I'm going to write down now. So we have the ribosomes, which are involved in translation. This blue string here that you see, this is actually the nucleoid region that contains the DNA. And the outermost layer, the pinkish one, not this purple one, but the pinkish one, is actually a cell wall and is a peptidoglycan cell wall, which the name says it all. It's comprised of peptides and carbohydrates, or proteins and carbohydrates. And two characteristics of prokaryotic cells that I think it's important to understand is that they're rod-shaped. We mentioned that before. They're rod-shaped in other tutorials. And the cell wall is quite rigid, and it's due to its composition. So rigid cell wall. Two key things. The second thing I want to talk about is the loss of the cell wall, which was probably the first thing that happened to this ancestral prokaryotic cell. And if you think about it, this may not be or seem like an obvious step for you, but consider the possibilities open to a flexible cell without that rigid and limiting cell wall. So this is a very important key in the evolution of eukaryotic cells. Really have this down. Now, you lose the peptidoglycan wall. There is a loss of peptidoglycan wall, which is a rigid and limiting structure in the cell. And the cell is left with a very flexible membrane surrounding it. This is the third step I'm going to talk about. And what's going to happen is this membrane is going to start folding inwards in a process called infolding, like I'm writing down. And also, you're going to increase the surface area, or the area surrounding the cell, which separates the internal environment of the cell from the external environment. And this will enable absorption of nutrients and gases, very important for the cell. So this is a very important step for this evolution. And with this, you're going to see a more rapid and efficient metabolism. So there will be an increase in metabolism or metabolic rates in the cell. So very important step right here. And with this, there is an important point to make as well, is that the surface area to volume ratio is going to decrease. And why is that? Because you're going to see that the volume is going to increase in a much higher rate than the surface area or the area surrounding the cell. And this has to reach a certain equilibrium, because if you have really large volume and a small area surrounding it, you're not going to be able to sustain the cell with enough nutrients and gases, because the area where they can come in is not enough for the size of the internal size. So the cell's volume has to reach a upper limit. So this is a very important, and this is where the surface area to volume ratio decreases. I also want to show you here where there is some inward folding. This is like an example I'm showing you here, and this is the area where it's pinching. So this is my infolding. So I'm going to use the same figure that I used before and to explain step number 4, where there is infolding happening or inward movement of the membrane, and internal membranes here are being formed. And they are surrounded by these structures called ribosomes, very important in translation. When you go from DNA or mRNA, we'll talk about that later, to proteins. And they're studied with ribosomes, and also they start to surround the nucleoid region where DNA is located. So some of these membranes are surrounding the DNA, and they're going to form an important structure that we're going to talk about later. I'm going to show you here, this is my inward folding of the membrane with these circles. These are my ribosomes. There you go. And another important thing, oops, sorry, just cleared it for a little bit. Another thing I want to talk about is a process, or something is going to happen. The flexible surface will start to pinch off bits of the environment and bringing it to the inside. This is a process called endocytosis, like I wrote here, and I'm drawing here. This is my endocytosis and ribosomes surrounding the membranes. Very important process that we're going to talk about later in other tutorials too. Now, at this stage of eukaryotic evolution, the cell is moving on to another stage. I'm going to say or call it the 5th step or stage, where this very complex framework is going to start to be developed. It's called the cytoskeleton, or the skeleton of the cell. And this framework is made up of actin fibers and microtubules, and it would allow a lot of things in the cell, and that's why it's a very important step here. I'm writing it down, actin microtubules being formed and gathered in the cell at this stage. And the functions, I want to write them down, because you're going to understand why is this framework being formed at this stage of the game. And you can clearly see this would allow the cell to manage to change shapes. One of the things, change in shape, or changes in shape. And the second thing, it will help distribute the daughter chromosomes during mitosis. So distribution of daughter chromosomes or chromatids, and this is during mitosis, or the formation, helps in the formation of that mitotic spindle that we are also going to talk about in a different tutorial. And another thing that is going to happen is that the cell is increasing in size. Therefore, it needs a network that helps transport or move things around its cytoplasm. And this framework, the cytoskeleton, will help or allow the cell to do that. And now I want to talk about the formation of the structure very briefly. Formation or development of the flagellum. This is a very important structure that eukaryotes have or possess, and it's actually part of the cytoskeleton as well. and helps the cell movement. This is extension here that I'm going to draw, and you're going to see the increase in size of the flagellum throughout the evolution of eukaryotic cells, and helps in cell movement, writing it down right here. Well, in step number 6, something very important happens. And as we spoke in number 4, I believe, where you see the infolding or inwards movement of membranes and formation of these unfolded membranes that you see here, which start to surround the nucleoid region where DNA is located in prokaryotes. So you can see here the area where DNA is being enclosed by these unfolded membranes. And this is what we call the precursor of the nucleus that we know today. And this is the structure that defines or distinguishes a eukaryote. A true eukaryote is the true nucleus. Because, as you know, prokaryotes do not have a nucleus. They have the nucleoid, a region with DNA, but not a defined or enclosed nucleus with DNA inside. And here's what you see, these membranes starting to gather and form what we know of today as the nucleus. And another thing that is important that I spoke about or talked about in number 5 is the development of cytoskeleton. And these 2 steps actually took us to the point of a first true eukaryote in the evolution of eukaryotic cells. So the formation of a precursor of the nucleus with enclosing of membranes surrounding DNA and development of the cytoskeleton takes you to this important step, number 6, the first true eukaryote. And notice how far you've gone since the first prokaryote when you lose that rigid cell wall, and now you're left with a flexible membrane that allows you to go through all these processes and take the cell to where it is now at this point. Now we are going from this structure to our first true eukaryote to a more complex one that we're going to see here, and I'm going to call this number 7. And step number 7, you can still see the flagellum growing and developing, this structure that enables propulsion and also movement in the cell. And another important thing that is happening in this new eukaryotic cell is that the membrane continues to fold inward, as I'm showing you here. And you can see some enzymes here. These are actually structures called lysosomes. Lysosomes. These structures are very important because they digest micromolecules in the cell. What is it? Of micromolecules. And they are digestive vesicles that evolved. So they evolved from the endoplasmic reticulum, from the earliest form of the endoplasmic reticulum. That's important to know. So the lysosomes are very important in cells as we know of today, and they start to form at this stage of the game. So in number 7, you can see that the inward folding continues, and there are enzymes here from the endoplasmic reticulum, the earliest form, that will help the formation of early lysosomes. And also notice that this cell is taking a shape of something that we call phagocyte. And the phagocyte are these really large macromolecules that are able to, and they're motile, they move, so they're motile cells. As you see, the cytoskeleton is present, so they become motile. They also are able to engulf. and able to digest other cells or other structures. And keep this in mind because this new structure, this new eukaryotic cell, will use these characteristics to move on to the next step. So keep that in mind. To go from this organism to a more complex one, step number 8, I want to talk about something that was very, very important in this part of the eukaryotic evolution, the eukaryotic cell evolution. And it's something called endosymbiosis. It's part of the title of our tutorial because it is quite important, or endosymbiotic theory. And symbiosis means organisms living together. And endo means inside. So these organisms are sharing nutrients, gases, they are having shared metabolisms, etc. So this is a form of endosymbiosis. And how do you get to this theory? How scientists come up with this theory? So at this point of eukaryotic cell evolution, you see, or millions of years ago, you see that there are a lot of these organisms on Earth called cyanobacteria, and they're great photosynthesizers. And as you probably know from basic biology, the product of photosynthesis is oxygen. So if these organisms are producing a lot of O2 and being released into the atmosphere, you have an oxygen-rich atmosphere at this point. And as you probably know, oxygen is quite harmful to organisms, especially if they don't have ways to cope with it or to metabolize it. It can be a quite harmful molecule. So this is how scientists start to question how were these cells, these early eukaryotes, able to cope with this new environment? How were they able to deal with lots of oxygen in the atmosphere? And this, remember when I talked about the phagocytic capabilities of the newly eukaryotes, where they're able to engulf new cells or bring in new cells into themselves? Well, this is what happened. There was ingestion and incorporation of a prokaryote or a prokaryotic cell that was able to deal with these harmful properties of oxygen. So the way cells thought, let's say, quote unquote, thought about dealing or coping with this new kind of environment was to bring in a helper, let's say, a cell that was able to deal with the O2 in the atmosphere. And this is important to know because now we're going to have a series of events that are based in this endosymbiosis theory or endosymbiotic theory, bringing in organisms to cope with this new environment. One of the first organisms to be believed to have entered the cell, become part of the cell, of the eukaryotic cells, were the peroxisomes that still exist. And these organelles have detoxifying properties. They're able to disarm the toxic products of oxygen that you see in cells, like that are results of oxygen metabolism. And one example is hydrogen peroxide. It happens in the cell. There are a lot of metabolic processes going on that produce hydrogen peroxide, and the cells have to be able to cope with this, and they do it through these nice organelles called peroxisomes. And you see here how they entered the cell. So one thing that I want to show here is another important endosymbiotic event. The first one we saw was the incorporation of the peroxisomes to deal with oxygen in the atmosphere. And this one, which I will call step number 9 of eukaryotic evolution, is the incorporation of a proteobacterium that evolved into what we know as today as the mitochondria, or a mitochondrium. Drium. and this is singular. If you want to talk about plural, you do mitochondria. That's plural. So this Proteobacterium that we see here that entered the cell through endocytosis and then adapt to it was ingested and incorporated. Endosymbiosis here is happening as well. And mitochondria became what we know today as the organelle responsible or that generates ATP. And ATP, as we know, is the energy of the cell. So after the incorporation of the Proteobacterium that became the mitochondrium that we know today, this is the step we consider that the basic evolution of the eukaryotic cell was complete. So here we can say it's complete evolution, incorporation of mitochondria. But I didn't want to finish this tutorial or finish talking about the evolution of eukaryotic cell without talking about the incorporation of these organelles that were actually cyanobacteria, or the organisms able to do photosynthesis that I mentioned before, so cyanobacteria. And these organisms, after being incorporated through endosymbiosis in the new eukaryotic cells, these become chloroplasts that we know. Probably heard about plant cells, which contain chloroplasts, not human cells. Human cells do not contain chloroplasts, very important, but plant cells have these organelles that are able to do photosynthesis, a process which is very important for plants and for humans as well, because here's how you produce the oxygen or that you see in the atmosphere. So you have an idea of how chloroplasts were formed, and I believe that's important to know, but maybe not for medical school, but in general, I think it's an important step of endosymbiosis as well, or the evolution of eukaryotic cell. But the end of it, I also want to mention and pinpoint how throughout these pictures you can see that the cell becomes more organized. You can see that the nucleus is becoming more organized, and the endoplasmic reticulum is being formed. Is being formed around the nucleus with these membranes that were folded inwards, the infolding that I talked in the beginning, and how the microtubules and the actin filaments are being organized more and more throughout this evolutionary process. So also, I believe you kind of notice throughout these pictures that they are becoming more and more organized and specialized, but it's good to pinpoint and to let you know that this is what's going to happen, or this is what is happening in the cell at this point. So you have an idea of how the evolution of eukaryotic cells happened at this point. So I will not do this in real time because you will be very, very bored if I have to write all these like in real time, but I will speed up the process. But I want to do here something that is go through the evidence that scientists use to prove that mitochondria, and other organelles aroused from bacteria or prokaryotes, like I mentioned previously. And there are many points, but I'm going to mention the ones I believe that are the most important ones. And the first one is that mitochondria and other organelles have this form of division, or the way they multiply or go from 1 to 2 through binary fission. So cell division is binary fission. It's the type of cell division you find in prokaryotes, because in eukaryotes you find mitosis. It's a different type of cell division. The second point is that they are surrounded by two or more membranes, and the innermost of these shows differences in composition from the other membranes of the cell. They are comprised of peptidoglycan wall remnants, same thing that you see in prokaryotes. Third point is that similar DNA to bacteria, the DNA you find in mitochondria, for example, is very similar in size, and it has the same circular shape like the DNA you find in prokaryotes or bacteria. Fourth point is that the organelles have ribosomes. So the mitochondria, for example, have ribosomes like those found in bacteria, and they're more or less 70 S units, so that's more or less the size of it, 70 S units, or Svedberg or sedimentation units. That's the units used to describe the size of the ribosomes here. And the fifth point is that they have similar structure. When you see chloroplasts, for example, and compare them to cyanobacteria, they have not only similar physical structures, but they do have similar functions like photosynthesis. The other point is that mitochondria have similar enzymes and transport systems as bacteria. That's another piece of evidence that shows you that they derive from bacteria. And the last point is that mitochondria and other organelles have similar size to bacteria. So keep that in mind. And these are the pieces of evidence that link all of this together. We just reached the end of this tutorial, and I want to give you a perspective, a general perspective of what we talked about of this evolution of the eukaryotic cells. And I want to mention 5 basic steps that we just talked about. Just do a very quick summary. The first one is the origin of a flexible cell surface when the first prokaryotic cell lost that rigid cell wall that enabled to have to be left with a flexible surface and to start other processes. The second one is the origin of the cytoskeleton when you start to form or developed actin and microtubules and a flagellum that enables cell shape, the change of cell shape and also movement. The third one is origin of a nuclear envelope, where we talk about these membranes that are a result of the infolding surround the DNA in the nucleoid region and form the precursor of the nucleus that we know. And here is when we have the first eukaryotic cell, true eukaryotic cell. And then the fourth happening or event is the appearance of digestive vesicles like the lysosomes. And the fifth one and final one is the formation of some organelles through endosymbiosis. And this was the last step I talked about, where I mentioned the formation of how cells developed the peroxisomes, mitochondria, and the chloroplast that we know today.